This is Key Assessment 3, Core Concepts for the Lower 6, Paper 2. Question 2. Long question this. In most eukaryotic genes, coding regions, exons, are interrupted by non-coding. Coding are exons, non-coding are introns. The entire gene is copied. So what you know about is making messenger RNA, and there's a pre-RNA, and we get rid of the introns. And then that's translated into the final structure of the protein. So reasonably straightforward there. Explain transcription, including the roles of the enzymes. So 3 marks. You need to talk about transcription. So just pretty straightforward. AO1, talking about unwinding of the double helix of DNA, complementary base pairing, explain that with maybe some examples, and mention the enzyme that joins nucleotides onto the growing messenger RNA chain. So pretty straightforward that. Explain why it's important that when RNA is finally translated, consists only of exons. So exons, I always remember exons are expressed in the protein. Introns, well, they're kept in the DNA. You're kept in, they're not expressed. So why do we only want these exons? Well, these introns, they could contain all sorts of different codes, and that's going to be, coding for amino acids. So our protein could be different, in a different shape. You just need to explain, what is going to get translated and what might not be translated. Explain how a change. This is much the same question, really. We explain a change in an intron. Well, it's not there, so it doesn't get translated. So we could change anything you like in an intron, and it wouldn't affect our protein. Whereas if we change an exon, well, that's going to change the codons on messenger RNA and change our translated, protein. So again, not too bad, pretty straightforward. We have got length of DNA, template strand, and then messenger RNA complete the correct sequence of amino acids. So it would help here if you divided these into 3s. There, into our triplets, and then you're just looking up these. So AUG, because this is messenger RNA, so that's messenger. So A, U, and then G. So that's, hang on, AUG, A, U, G. There we are. So it lines up with the A, the U, and that final G there. So methionine, and you're just doing that as you go along. Mutation, you can do that as well. Explain why it's a silent mutation. Well, hopefully, even though this is different, it produces the same amino acid. You're working out some ratios. I've been quite lenient here with working out which one you divide by what. So the key here is making sure you do the right one. So follow the instructions. Calculate the ratio to 2 decimal places for a human sperm. So we are doing that one. And a ratio, remember your surface area to volume ratio, things like that, then it's something to 1. So adenine to thymine, we need to turn thymine into 1, and you turn anything into 1 by dividing by itself. So if A is 30.7, T is 31.2. Well, what do I do to 31.2 to turn it into 1? I go 31.2 divided by 31.2, and that's 1. So if I divide that by 31.2, that equals 1. And I just need to do the same to this side. All the time in biology you're doing the same thing. We did it in the last question. Do the same to both sides. So divide that by, so 30.7 divided by this one over here, 31.2. And that is 0.9839743, and 2 decimal places. I go, do I round that? So that's going to be 0.98. And expressing a ratio 0.98 to 1. So that's the way. If you do it the other way around, they'll let you have that as well. So, but it's traditional to do something to 1 in a ratio. And then you have the last part of the question. We have got some things from Pauling, some information from Pauling, and some information from Franklin. Your basis of this question is that you know what the double helix, you know it's a, the structure of DNA is a double helix, you know the sugar phosphate backbone is on the outside, you know the bases are on the inside and complementary bases, and you know it's a double strand, you have 2 strands. So that's the information that you need. And then all you need to do is go, do I agree with these statements or do I disagree with these? So it's unlikely that sugar groups are in the core. Well, what do we know about that? Sugar's in the core. No, the bases are in the core. So we're going to say, yeah, I agree with that. And so on. And one from over here. We've got phosphate groups on the outside. And you know that the phosphate groups are on the outside, sugar phosphate background. And I say, yeah, I'm going to agree with that. So you're just picking out a couple of things from each one. We have 4 marks, so we need 2 points for each one. Good luck.